10th standard ICSE Biology, concise Chapter 2, Structure of Chromosomes, Cell Cycle and Cell Division. In 9th standard, all of us have studied that every cell, be it animal, plant or a microbe, has a nucleus, and the nucleus has chromosomes. The chromosomes are made up of DNA, which is the genetic material of every cell. It has genetic information, which is passed from the parent to the offspring. It's a brilliant way to pass on the information so that the child is exactly a replica of the parent or a mixture of the parents. Well, I am assuming that you studied 9th biology seriously to remember this. But what are chromosomes? Well, literally, chroma means colored and soma means body. So when the microscopes were invented, the cell was observed and studied, but we couldn't see all the organelles yet. One way of seeing various parts is by using dyes. Now, chromosomes take on particular color with the help of some dyes, due to which they were discovered. So chromosomes are made up of a material called chromatin, and we'll study more about it shortly. This chromatin was first seen under an electron microscope, which is a very powerful microscope, and it was extremely thin, long, and darkly stained fiber, hence the name chromosome. You see, when cell division occurs, the chromatin fibers, they condense into organized chromosomes. And when the cell is not dividing, the chromosomes relax and the fibers become chromatin fibers once again. So chromatin fibers and chromosomes are made up of the same thing, that is DNA and histones. So we have a definition of chromosomes. They are highly coiled ribbon-like structures. They were first discovered by Walter Fleming. He was studying the cells of the larvae of salamander, an amphibian, can live in land and water both. Now whatever I've marked as read, that you just have to read, you don't have to learn it. Very unlikely that anything will come in the exam from here. And the introduction of every chapter is, of course, again, just read. Chromatin, another definition. What is it made of? 40% of it is DNA and 60% are proteins called histones. Let's look at figure 2.5. So here we have a DNA strand. We'll study this structure in detail shortly. This DNA fiber is a double helix. There are two of them. They are coiled around a core of 8 histone proteins. So together, this is called one nucleosome. Many such nucleosomes coil together and supercoil, just like the coils of a landline telephone. If you've ever felt irritated by this, you would remember. And the supercoil structure is called chromatin fiber. That's what is present in the nucleoplasm of a nucleus. Now, during cell division, this chromatin fiber again condenses a lot, and now it is called a chromosome. Now, a chromosome may have a single chromatid. Chromatid is different from chromatin, all right? Chromatin is the material, but this is a supercoiled chromatid. Or a chromosome may have 2 sister chromatids attached to each other at a point called centromere. Now, the sister chromatid is an exact duplicate, a replica of the other chromatid. That is because during cell division, it's possible that they will get separated and each new cell should get the exact replica. So we will study these processes in detail in this chapter. Well, the shape of the DNA molecule was first studied by Rosalind Franklin in 1953. And based on her research, later Watson and Crick finally worked out the double helical structure of the DNA, the very famous diagram of the DNA, one of the most beautiful diagrams in biology. And for this, they were awarded the Nobel Prize, Watson and Crick. Rosalind Franklin did not win the Nobel Prize. I guess this is because of misogyny. Now, a single DNA molecule is very large, hence it's called macromolecule. It has 2 complementary strands, a double helix, and these rungs of the ladder, the steps, they are made up of nitrogenous bases. So in figure 2.3, bravo, double tick it, which means that you have to practice it because drawing can come in exam. So how many strands do you see? I see 1 strand and 2, so 2 strands. Each strand is made up of few components. They are called nucleotides. Let's study the structure of one nucleotide. Let's zoom in. Each nucleotide is made up of a phosphate group, a pentose sugar, and a nitrogenous base. So these 3 components make up a nucleotide. So how many nucleotides do you see here? That's right, 6 nucleotides. And notice how this strand is running opposite to the direction of this strand. Notice how the pentose sugar is pointing upwards here, but here it is downwards. Yeah, you have to keep in mind these things while drawing it. And the nitrogenous bases are so complementary that A will always bind with T and G will always bind with C. There are only 4 types of nitrogenous bases in a DNA strand: ATGC, adenine, thymine, guanine, and cytosine. Now, they are connected with the help of hydrogen bonds. Between A and D, we have two hydrogen bonds, and between G and C, we have 3 hydrogen bonds. And this is the code language in which all the information about you is stored in your DNA, which you got from your parents when you were conceived around 14 years ago during fertilization. Do you remember? You got 50% of your DNA from your mom and 50% from your dad. And everything about you, well, most of your physical features were predetermined even before you were born. This chemical code language holds the secret of how you would grow. Your height, your skin color, the color of your iris, will you be good at biology, is all written in your DNA. But hey, you are what you are not just because of your DNA, but also because of environmental factors. So don't just blame your genes for being poor in some subject. Work hard and you can excel in anything. Another fun fact: since all life has evolved from a common ancestor billions of years ago, we share a lot of our DNA. In fact, all human beings share around 98 to 99% of DNA. In fact, 50% of your DNA is similar to that of a banana. Go figure. So here you can see that they're not coiled with each other. Once they coil with each other, they look like this. You see, this code is actually a recipe. You know who are the protein synthesizers in our cell? Yes, they're ribosomes. What, you forgot? You had studied this in 9th standard. Ribosomes need the recipe while making proteins, and they get that recipe indirectly from the DNA. They use this to link amino acids with each other to make so many different types of proteins. And these proteins ultimately make a lot of things in your body: your enzymes, your hormones, pigments, and ultimately these things affect everything about you. Even before you were born, when you were just a tiny embryo with no real physical features, how did the cells know that you have to grow into a human being? I mean, it could have grown into a mango tree, for example. I know you wouldn't have preferred that, but how can a single cell know that after repeated divisions we have to grow a head, hands, heart? Well, they take the instructions from the DNA. And if there is a flaw in it, which is called a mutation, mostly it is corrected by our body. We have a nice system, but sometimes mutations may lead to disorders and even cancer. That's why I stay away from carcinogenic things like smoke, alcohol, radiations, pollution, etc. Histone proteins mark this as a function. As I explained, that they are proteins and they help the DNA to coil around it to form nucleosomes. Lots of words are similar, which can be confusing. How will you remember it? Well, you can revise again and again, and you can write things down, at least the main points and main words. That way, your spellings become stronger, your handwriting improves, and you remember for a long term. Now we have another definition of nucleosome, mark it. We have 1,000,000 nucleosomes in every cell. In fact, in every chromosome. And every human cell has 46 chromosomes. 23 are replicas of your mother's chromosomes and 23 from your father. 23 pairs. Now, this part is entirely read, it's irrelevant. This speech box is a very good summary, good to revise. We have 2 kinds of nucleic acids in a syllabus: DNA and RNA, deoxyribonucleic acid and ribonucleic acid. RNA is useful for protein synthesis. They are single-stranded, not double helix like DNA. Ribosomes take instructions from RNA, and RNA takes instructions from DNA. Now, the chromosomes are made up of DNA and histones, as we had studied. Nitrogenous bases are of 4 types in DNA. But in RNA, we have a new type, uracil. So instead of ATGC, they have AUGC. RNA does not have thymine, it has uracil. Remember that A and G are purines, whereas TCU belong to a category called pyrimidines. So a purine always links with a pyrimidine, they're complementary. And remember the difference between nucleotide and nucleosome. Remember the name of the scientists as well. Now this is figure 2.6. Single tick means you should study it. Diagram based questions can be asked, labeling can be asked, but drawing would not be asked. As I told you, the 2 sister chromatids are linked with each other at the centromere, which need not be at the center, it could be anywhere along their length. And they are duplicates of each other, exact. So we have a definition of chromatid, a single chromatid. One vertical half of a duplicated chromosome is called a chromatid, and two of them connected by a centromere are called sister chromatids, which will get separated during a cell division phase called anaphase. We will study all the phases of cell division later. So what is the function of centromere? Well, to attach the chromatids, and also during cell division, it will help to detach both of them. They will get separated from each other and they will be pulled apart from each other. Now I told you that the DNA has ADGC, which is nothing but a code, a recipe to make proteins. So every part of a DNA which has the code for one particular recipe is called a gene. Yeah, the DNA has many genes, and a gene is made up of many nucleotides. And it is by reading the genes, let's say CG, is one particular gene, which is a recipe to make one particular protein by the ribosomes. Now, how is that translated and how is the protein manufactured is in 11th standard syllabus, and it is a fascinating process. It is mind-blowing, one of my favorite processes in biology, entire biology. There are many short, animated, simple videos available on YouTube. Just type conversion from DNA to protein. In fact, let me show you a glimpse of one such video. Notice how the complementary nitrogenous bases are being attached. And now this is a ribosome, and see how it will use the RNA information to make proteins. So every gene is a recipe to make a particular kind of protein, and ultimately proteins control our body. So that's how DNA has all the information of our body. So mark the definition of genes, and definitions should be learned word to word. Yeah, you should understand first, but don't try to write in your own words because you might mess up the meanings. There are technical words which are essential for an accurate definition. Genes are fascinating. It has opened up a new branch of science, genetic engineering. What if we could hack into the genes, and once we've understood the code language, we can change the recipe, we can make new proteins so that right from the embryo stage, we can give properties to the life, to the living organism, which would make it superhuman, like give it wings or give it ability to do photosynthesis. We just have to learn the DNA of other organisms and learn how to hack it, cut, copy, paste into our DNA. This genetic engineering will cure diseases, give us bumper crops, create mutants, and, well, have superheroes. Not the lame ones which Marvel has, the real strong superheroes like DC ones. Whatever is not in syllabus can be ignored, but you might find it interesting to read, so go ahead. You might even find it useful because DNA fingerprinting is basically to compare your DNA with that of your parents. Often you might have some doubt in your mind when you see how dumb you are compared to your wise parents, where you exchanged at the hospital or who knows what. So you might just go in for it. And if you do take up biotechnology and genetic engineering as a career option, because that is the most happening field in 21st century, apart from AI, of course, well, don't play with genes. Don't try to experiment with life because who knows, you might accidentally create Godzilla. Now, why is cell division so important? Why do we need a continuous supply of new cells? Well, there are four reasons. First, growth. We grow, and as we grow, we need new cells. At least till the age of around 18, you grow vertically. After that, we may grow horizontally when a tummy comes out. What is happening even now? Fair enough. For replacement, yeah, wear and tear happens. Like our RBCs have a lifespan of 120 days. So every day we are losing thousands and millions of RBCs, and new ones are being born. For repair, sometimes we get injured. So if you get a cut in your skin, you may have noticed that new skin develops after a few days out there. That's because of cell division. New cells are being born, and these new cells are replicas of the surrounding cells. They have the same 46 chromosomes. Each and every cell of your body has the same 46 chromosomes. Remember, the cell division which happens in these three cases is called mitosis. They all have 46 chromosomes, at least in human beings. If you have 48, then you're a gorilla. And they are simply copies of your parents' DNA. 23 chromosomes from your mom and 23 from your dad. But there is another kind of cell division called meiosis. Now that takes place only for the purpose of reproduction. Now I see that you become more attentive. Reproduction in humans is sexual reproduction, so two parents are required. But if each parent gave 46 of their chromosomes, then the zygote formed will have 92 chromosomes, and that won't be a human being. So the male gamete, that is a sperm, has only half the chromosomes of the father, 23. And the female gamete, again, has only half the chromosomes of the mother's cell, 23. And when the fertilization happens, then the zygote has 46 chromosomes once again, which then multiplies. One cell becomes two cells, two becomes four. Very soon an embryo is formed, and each cell has 46 chromosomes, and mitosis continues. So meiosis happens only when gametes are being formed. In meiosis, the number of chromosomes are halved. In mitosis, which happens for a growth, the number of chromosomes remain the same. So in mitosis, which we are going to study in detail in this chapter, it's called equational division. Number of chromosomes remain the same, 2n. We have 23 pairs, and it's called the diploid condition. Whereas meiosis, called the reductional division, which we won't study so much in detail in this chapter, it's called reductional division because the number of chromosomes are halved. The daughter cells, yeah, they're called daughter cells always because the cells again can multiply further. That is the sex cells, sperms and eggs, they have only 23 chromosomes, so N, that is haploid condition. Now what is a cell cycle? Double tick figure 2.8 and practice it for homework. It has a few steps. Let's start with G1, the first growth phase. A newborn daughter cell, small. She has to grow. She has to fill in the shoes of her parent. So what happens here is RNA and proteins are synthesized. Because she's small, she got only half the RNA and proteins from the parent. The mitochondria were also halved, so now she'll have to multiply that. The chloroplasts also have to divide, but that will happen only in plants. If you remember, mitochondria and chloroplasts have their own DNA. That is why they are able to divide on their own. They are semi-autonomous. In fact, there is a theory that billions of years ago, before life as a cell started, mitochondria and chloroplasts were separate organisms. They could replicate on their own, and that is the definition of life. The only thing which differentiates us from non-living things is that we can reproduce. There is no other difference if you think about it. So mitochondria and chloroplasts can divide themselves and they can produce their own energy, ATP, to survive. But during evolution, they came together to form a team called the cell, and now they're dependent on the cell a little bit. They can't live independently anymore. You see, once you are bonded with a team, it's difficult to become independent. So be careful about what team you join. So after the first growth phase, the cell has a decision. Either it can go into a resting phase and do its functions as a liver cell, a brain cell, a red blood cell, a skin cell, or it can multiply further. If it decides to multiply further, and hey, everybody has their own choice, okay, whether to reproduce or not. You should not be pressurized into it. You choose your time. You want to focus on your career or you want to reproduce further. If you do, then don't just reproduce at any moment. First, make sure you have enough resources to give to your daughter cells. You have enough RNA and proteins and DNA, because when there will be separation of the cells, the daughter cells should get enough material to stand on their own feet. So there is a synthesis phase. More DNA is synthesized and the chromosomes duplicate, but they remain attached. And what are they called now? The sister chromatids. And where are they attached? At the centromere. So you know that you're going to have two daughters. So let's multiply everything by 2 so that when we separate, the daughters get 46 chromatids. This is mitosis, by the way. And after this, we have a G2 phase, or the second growth phase. More RNA and proteins are synthesized. And then we finally have mitosis, which has further phases, which we'll study in detail in the next section. This entire process, G1S and G2, is called interphase. And after interphase, we have the mitosis, in which we have two daughter cells, and the cell cycle continues. This is explained in detail in all this theory which you have to study. This paragraph is just read, just add one word here, plant. Now, the formation of new DNA, how does that happen? Look at this process. First, you have a double helix. It is wound together. Now, there is an enzyme which will break the bonds, the hydrogen bonds, and will unwind into 2 single strands. And with each strand, a new strand is being formed. So ultimately, we have 2 double helices, which we saw briefly in the animated video I showed you. So that's how DNA is duplicated. Remember, here we had A and here we had T. So we'll have a new T with the A and a new A with the T. So both of them are mirror images of each other. Can the cell cycle go on endlessly? No, it will stop. You see, when you photocopy some page of your book, the photocopy won't be exactly like the page. There will be some errors here and there. What if you photocopy the photocopy again? The quality will reduce, and there will come a time after multiple photocopying that the quality will deteriorate so much that it will become useless. So a cell also has limitations. Nobody can be immortal. The cell will divide and divide, but after decades and decades, and say 100 years, the cell division will just stop because now the DNA faults have accumulated so much that either you will have cancer and die, or you'll have some other disorder and die. But that's just a transition phase into a universal enlightenment. Now, different cells of a body have different lifespans, and how beautifully they just live harmoniously so that you can exist. And they don't mind dying for you, like WBCs. They kill the germs and die in that war, but they make sure that you survive. So take care of your cells, eat healthy. Cells of the island's brain and some nerve cells, they can't multiply because they've lost the ability to do cell division. Skin cells, they multiply regularly. I mean, you lose skin cells every day. Example, dandruff. So learn the lifespans of various cells. Liver cells, well, they take 1 to 2 years to replicate, so don't destroy them. You know how. In plants, not all cells can divide. Permanent tissue can't. Meristematic tissue can. And the cells in the ovaries and testes, called germinal cells, they can divide, but they undergo a very special type of cell division. What is that called? Meiosis. Now, what if the cell cycle is disturbed, the DNA is damaged because of gamma radiations, etc.? There'll be mutations. And what if the cell cycle goes on continuously, uncontrolled manner? A tumor is formed. And the tumor can be benign, harmless, but it can also be fatal, and it can become cancerous. It becomes like a separate entity. It no longer cares about you. It just wants to grow and grow, consume all the resources and the nutrients from your body. It doesn't realize that if you die, even it will ultimately die. It just keeps on growing. As we grow old, the number of new cells produced runs short of those that are dying. There are some special cells inside a body, like in the bone marrow, called stem cells. Stem cells are very useful even in the embryo because they can adapt to any situation. They can become any kind of cell. If they're introduced into the liver, they will become liver cells. And if they're introduced into skin, they'll become skin cells, and so on. Hopefully, we will master this technology and cure diseases like brain diseases like Alzheimer's, etc. And the next topic is cell division, which will be taught in the next video. Hi students, this is AJ sir. If you found this video useful, press the like button. Also, if you'd like to enroll for my test series or online lectures, email me or message me on Instagram. Check my description for more information.